Dear student, today we will be talking about cloning vectors in gene transfer methods. The foreign gene, which is the gene of interest, is transferred to host to get it multiple copies. This process is called gene cloning. To do this, the foreign gene is first loaded on vector. We say this is vehicle or cloning vector. This result in the generation of rDNA, recombinant DNA, or we also say chimeric DNA or hybrid DNA. Then second step is this rDNA is introduced into the suitable host where the foreign DNA along with the vector DNA replicate. That means it form multiple copies. For this, various techniques are used. So how to introduce this DNA? First, the DNA is loaded by using the vector, okay, and then this DNA is introduced in the host. So there are certain techniques by using which we introduce this DNA in the host. First, let's talk about the cloning vector. Now understand the definition. The cloning vector is a small piece of DNA. This DNA could be of virus or bacteria that can be stably maintained in an organism which is known as host, and into which the foreign DNA fragment, that is the gene of interest, can be inserted for cloning purpose. So this is what we mean by the cloning vector. Now let's look at the classification of vector. This is based on your syllabus. We have natural vectors which uses plasmid, bacteriophage such as lambdaphage. Reconstructed vector out of these plasmid, we have PBR322 which is reconstructed plasmid, cosmid and phagemid, which are hybrid between the plasmid and phage. Then we have artificial vectors designed for eukaryotic genes. um, Only we will discuss YAG, that is yeast artificial chromosome. We also have bacterial artificial chromosome and many others. Okay, so now we'll come to the characteristic of cloning vectors. So what are the essential features of these cloning vector? A cloning vector should have what all things? So first important thing is the origin of replication. Second, presence of selectable marker. Third, multiple cloning site. And fourth, the size matters. Size should be less. Now, origin of replication are the DNA sequences at which the replication is initiated. The cloning vector contain relaxed replication control so that they are able to produce multiple copies in cell. Now, what do you mean by relaxed replication? Means they can be independently, or you can say autonomously, have the capacity to initiate the replication, and their replication is independent of the replication of host chromosomal DNA. Then once they initiate, then they are using the host machinery to do the elongation and the termination. Selectable marker are required for the selection between the transformed and non-transformed cell, host cell. So transformed are the one which carry the recombinant DNA. They are also known as recombinants. And non-transform are the one which do not carry the recombinant DNA. They are known as non-recombinants. Those cells are called non-recombinants. Now, there are different types of marker. One of them is antibiotic resistance marker gene, such as ampicillin resistance gene, tetracycline resistance gene. Then second is use of lexizine, which codes for the enzyme beta galactosidase. So we will see how they are used as selectable marker, and they impart the selection between the transformant and non-transformant. Multiple cloning sites, they are required to insert the foreign DNA. These contain the restriction endonuclease target sites. Multiple cloning sites, they contain this target site. And next feature is the size of the vector. It should be less than 10 kilobase pair, because less is the size, more uh, will be the efficiency of transformation. So we will discuss next the types of vector. First, let's discuss now plasmid as a vector. It is double-stranded extrachromosomal DNA molecule, circular, self-replicating, and do not contain any vital gene for bacteria, means the genes which are required for the cell division. So what are the advantages when we are using plasmid as a vector? Small size, easy to handle. It carry antibiotic resistance as a marker gene, which impart easy selection between the recombinant and non-recombinant. Then third, useful for cloning small DNA fragment which are less than 10 kilobase pair. So the disadvantage is we cannot clone the large base pair DNA. Here I'll also talk about the TI plasmid, which is present in a bacteria called Agrobacterium tumefaciens. Okay, and this TI plasmid actually uh, contain uh, tDNA, which is called transfer DNA, and in natural condition, this bacteria can infect the plant cell, and it can introduce the bacterial DNA into the plant cells. So since this property is present with agrobacterium, so its plasmid is engineered to transfer the gene in plants. We will talk about this later on with example how it is used. So therefore TI plasmid is used to transfer the foreign gene in plant. Then we have bacteriophage. They are the viruses that infect the bacterium and multiply within them by using the bacterial machinery. So bacteria are the host. Okay, so lambdaphage is 49 kb size DNA, kilo base pair size DNA, and that is used as cloning vector. So you can use bacteriophage, simply bacteriophage as a vector. So let's understand what all things, how it is how it is structured. The double-stranded linear DNA has a stretch of 12 nucleotide at its either end, which act as a sticky and called cohesive and or cos sites. So this 12 around 12 nucleotide DNA, which is present at the extreme and constitute the cos sites. The cos site help in packaging of linear DNA to circular form so that they can easily fit in the protein coat. Secondly, these cos site also help to do the replication in circular form, and also to integrate the bacterial DNA within the phage DNA. Okay. Now, so this has a lot of significance. Now, if you see, the bacteria is infected by phage DNA. Okay, so bacteria have their own DNA, phage DNA is there, cos sites are there. So the DNA will enter the E. coli, and E. coli machinery is used to multiply the phage DNA. So many copies are formed. Now there are two different modes which are possible. One is that multiple copies are formed, and further these are encapsulated by protein coat, and the viral particles are released from the bacteria, and then they are ready to infect the next you can say bacteria. So this constitute the lytic cycle. Okay, if you see the other part, it may happen that phage DNA is integrated with bacterial DNA, and what will happen, this condition is called prophage, and then this DNA, number of copies of this you can say the hybrid DNA uh, between the bacteria and phage is formed. It is not like that ki whole of the E. coli is integrated with phage, but the any fragment of E. coli is integrated with phage DNA. So then they have to enter another cycle called lysogenic cycle, and Lysogenic and lytic cycle may alternate with each other. So the bacteria which is present in the, the phage which is present in the lysogenic cycle may enter the lytic cycle. Okay. Now, if you look at the lambda phage, if it is used as a vector, what are the important or you can say advantages? It is useful for cloning the large size around 10 to 23 kilo base pair of DNA, and then it is easy to detect based on the formation of plaque. So the virus, when they grow on the agar plate, they forms the holes which are known as plaques when they infect the bacteria. Okay, so if the plaques are cloudy, they are non-recombinant, and if the plaques are clear, they are recombinant. Now disadvantage is that it's very difficult, or it's not very easy to handle the bacteriophage. Restriction sites are large in number. So if you are using any restriction site or enzyme, then it is going to fragment the DNA of phage into so many pieces. Okay, so reconstruction of phage, or you can say engineered version of phage, is nowadays used. So in that, what is done is lot of unnecessary genes which are not required by the phage are removed. So around 25 to 35% of lambda gene are eliminated to increase the size of insert. Insert means foreign DNA, because right now its capacity is 10 to 23. So if you eliminate more gene from lambda phage, then more will be the size of insert that could fit into this DNA. Okay, so most of these genes lie in the lysogenic phase. These are non-essential region. So the capacity now has increased, and it can clone up to 37 to 15 kilobase pair of DNA. The mutagenesis is used to modify certain restriction sites, like for example, EcoRI site is GAATTC. So that could be changed to GGATTC by mutagenic agent, and now this is not recognized by the enzyme. So you can block certain restriction endonuclease sites so that the DNA is not fragmented by use of restriction endonuclease. So in the next video, I'll be discussing about the next constructed plasmid PBR322. Thank you.